So we're going to cover the taxonomy of viruses and also group 1 viruses in this lecture. So viral classification is pretty hard to do because we don't have a lot of information on where viruses actually originated from, and we don't have any fossils of viruses, so we don't know their evolutionary history very well either. So taxonomy of viruses and classifying them, you don't need to memorize this, but they go through the same order, family, genus, just like you do with cells. And then a viral species is just defined as a group of viruses sharing the same genetic information and the same host. And this is just an example of classification of the mumps virus and the measles virus. So they're actually fairly closely related. They're actually in the same family. Classification of viruses is based on a lot of different characteristics. So we can classify them based on their nucleic acid type. So whether they're using double-stranded DNA, single-stranded DNA, double-stranded RNA, or single-stranded RNA. We can also classify them whether they have an envelope or not, capsid symmetries, so are they helical in shape, icosahedral, or complex, and then also the size of the virus and the capsid could be used. But we're going to be looking at the classification system called the Baltimore system. So it was developed by a scientist named David Baltimore, and he organized the viruses into 7 different groups based on what type of nucleic acid they're using and then how they replicate their nucleic acid. So here are the 7 groups in the Baltimore classification system, and we'll go through each of these groups. But you don't need to memorize like group 1 is double-stranded DNA and group 2 is single-stranded DNA. I won't ask you that at all. So as far as just some generalizations for animal viruses, viruses that are using DNA as their genetic material are going to replicate the viral DNA in the host cell nucleus and then make their capsid proteins in the cytoplasm. So they're very similar to what cells do with their making their DNA and proteins. But if you have an RNA virus, they're going to replicate their RNA and make their capsid proteins in the host cell cytoplasm. So they usually don't need to go into the nucleus. So to start with this slide, this is really important to understand. We're going to be talking about plus strand or positive strand and minus strand or negative strand. So it's all going to come to comparing things to the viral messenger RNA, which I have a picture here of the viral messenger RNA up here on top. And we're going to say that the viral messenger RNA, another name for it is plus strand RNA. So you can see the plus strand RNA is the exact same thing as viral messenger RNA. So viral messenger RNA and plus strand RNA are the same thing. And then if you have a negative strand of RNA, here in the red, notice that it is anti-parallel. So it's running 3'5 to 5' compared to the viral messenger RNA, which is running 5 to 3'. And also notice that the negative strand RNA is complementary to the viral messenger RNA. So you have G's and C's and A's and U's that match up. We can also have plus and negative strand DNA. So the plus strand DNA here in purple, notice that it's running the same direction, 5 to 3' as the viral messenger RNA. And it's also the same sequence as the viral messenger RNA, except for instead of U's, there's T's. Negative strand DNA is anti-parallel, 3 to 5' compared to 5 to 3' on the viral messenger RNA. And also because it's negative strand, it's complementary to the viral messenger RNA. So you have G's and C's and A's and T's together. So our first group of viruses, this is the group 1, and these are double-stranded DNA viruses. And these are just some examples of some double-stranded DNA viruses. We're not going to talk about all of them right now, but we'll get to some of these more later on when we get to diseases. So with our double-stranded DNA viruses in group 1, most of the bacteriophages that infect bacteria belong in this group. And as far as viruses that infect humans, some important ones are the herpes viruses, pox viruses, which include smallpox, adenoviruses, which are one of the causes of the common cold, and then papillomaviruses, which can cause warts, and then also the human papillomavirus also will cause genital warts too. These double-stranded DNA viruses, because they have the same genetic material as what cells are using, they're going to be able to use a lot of the host cell machinery and not have to bring a lot of their own enzymes along. So this is just a picture of how group 1 viruses are going to replicate and make more viruses. So remember that viruses are made out of two things. So they're made out of their nucleic acid, which in this case is double-stranded DNA. And then they're also made out of the viral proteins, which make up the capsid. So in order to make new virus, you just need to make two things. You need to make more of your nucleic acid, which in this case is the double-stranded DNA, and you need to make more of the viral proteins to make the capsid. So in order to get from the double-stranded DNA to make the viral proteins, this is the same as what cells do. So these viruses are going to be using the host cell enzymes because they're the same as what our cells use. So we're going to take our double-stranded viral DNA and we're going to transcribe it. So when we transcribe it, remember we make messenger RNA, but remember we said that the viral messenger RNA is the same as plus single-stranded RNA. Okay, because remember that messenger RNA is always single strand, and it's a plus strand because that's just by definition the viral messenger RNA is the plus single-stranded RNA. So the enzyme that we're going to use to make the messenger RNA for the virus, remember the RNA polymerase is what does transcription. But we're going to add a term in front of the RNA polymerase. So the RNA polymerase is depending on the DNA in order to make that RNA. So we're going to call it a DNA-dependent RNA polymerase. And then once we get our viral messenger RNA, or plus single-stranded RNA, we will then translate that with the host cell ribosomes to make the viral proteins. And then those viral proteins will then form the capsid. The second thing we need to do is we need to make more of the viral double-stranded DNA. So we're just going to use the host cell enzyme DNA polymerase. So remember that DNA polymerase makes DNA, so that's what we're making here. And then if we look back, we're depending on the DNA for our instructions to make this new double-stranded DNA. So the enzyme, instead of just being DNA polymerase, we're going to call it DNA-dependent DNA polymerase. And then once we have the viral nucleic acid and the viral proteins, they will just self-assemble during the maturation phase of viral multiplication and just self-assemble. So what you need to know for the quizzes and the exam is that I will give you a drawing like this, but just leave some things blank and you have to just fill in the missing blanks. So you don't need to memorize this and be able to draw the whole thing out for me. So all you need to do, for example, maybe I have blank this enzyme here. So you would need to fill in, if I have a blank there, saying that's DNA-dependent RNA polymerase. Or maybe I have a blank here and you have to fill in what's being made. Well, that's plus single-stranded RNA. And maybe I have blank here, the process, which you'd have to fill in, oh, that's translation that's being done. So that's what kind of questions I would ask about this. So you don't even need to know it's group 1. You don't have to be able to draw this whole thing out. I will draw most of it out and just leave some things blank that you just have to fill it in. So here's an example of a group 1 virus, a bacteriophage called T4, which you do not have to memorize. I just wanted to give you an example and just kind of a nice review. So is this a virulent or temperate bacteriophage? So which cycle is it doing? Is it doing the lytic cycle or the lysogenic cycle? So this would be a virulent bacteriophage because it's coming into the bacteria, injecting its nucleic acid, and then it's immediately making new virus and then lysing the cell and releasing more virus. And all of this amazingly only takes 22 minutes, which is pretty fast. Okay, and then this is just another group 1 virus, another bacteriophage, bacteriophage lambda. So is this a virulent or temperate bacteriophage? So this one will be a temperate bacteriophage because it will infect the bacteria and then do the lysogenic cycle first, and then when it senses the bacteria is not doing well, it'll cut itself out of the bacterial chromosome and then do the lytic cycle, make new virus, and go find a new bacterial cell to infect. A very important group of group 1 viruses that infect humans are the herpes viruses. There are actually 8 different herpes viruses that infect humans, but I've only got 5 of them listed here. So the first one, herpes simplex virus type 1, that one most commonly causes cold sores. And then herpes simplex virus type 2 most commonly causes genital herpes. But it is possible for them to get in the other location, and so herpes simplex virus type 1, if it's on the mouth, can cause cold sores, but if it gets in the genital area, it could cause genital herpes, so they could switch places. The varicella zoster virus, that causes chickenpox and shingles when you're older. The Epstein-Barr virus causes mono and can also cause cancer. Remember, it's one of the oncoviruses. But remember, it's only going to cause cancer if you also have malaria as well, which that's not going to happen in the US. And then cytomegalovirus we'll talk about later. That's another common herpes virus. Most people don't know that they're infected, but if you do get infected while you're pregnant, it can cause birth defects and miscarriages. So herpes viruses are envelope viruses, and if you take a look at the picture here on the bottom right, you can see the capsid is an icosahedral shape, so it has those 20 triangular faces on it. The target cells for a lot of the herpes viruses are epithelial cells and neurons. So the 2 target cells for a lot of herpes viruses are epithelial cells and neurons, although some of the other herpes viruses do infect some of the white blood cells instead. And then here's just a couple of pictures looking at the herpes viruses. Here's a nice electron microscope view. You can actually see the envelope, and you can see the spike protein sticking out that they use to bind to the receptors on our host cells to get inside. And then you can see the capsid inside. And inside of that would be the double-stranded DNA. So if you get infected with a herpes viruses, there's 2 infections that occur. So the productive or primary infection, that's actually the same thing as a cytocidal or acute infection. So the cell ends up dying, and then that will cause the symptoms. The latent infection, remember latent means hidden, so the immune system will start clearing away the herpes viruses, but it doesn't quite get all of them, and some of them will go and hide out in cells like the neurons. The herpes virus will go dormant. You won't produce any new virus. It's not detectable. And then if your immune system becomes suppressed later on in life, the herpes virus can start making virus again and then cause another infection. Here's a picture of herpes simplex virus type 1 that causes cold sores. So when you first get infected, the herpes virus will infect the epithelial cells that are around your lip, and they will cause an acute or cytocidal infection, and so they cause the cold sore. As the immune system is getting rid of the virus, clearing it up, the cold sore is resolving. Some of the herpes viruses will invade the sensory neurons that are around the lip, and then they will migrate up to the neuron cell body, and then they become dormant there. And then if you're stressed or sick, immune system is down a little bit, then the herpes virus can crawl back out the neuron, come back to the lip, and reinfect those epithelial cells and cause another cold sore. So that's a latent infection that's been reactivated. Okay, and then we just talked about all of this, and just the only thing just to mention about this slide is just because these herpes viruses have an envelope, remember that they get the envelope from the host cell. So remember that they can get it either from the host cell membrane or from one of the internal organelles. So I won't ask you that the virus envelope is generated from the Golgi apparatus. I won't ask you that, but just know that the viral envelope comes from the host cell. Okay, and you don't need to know this picture either, but it's just showing you the life cycle of the herpes simplex virus type 1 infecting an epithelial cell here. Okay, and then we talked about the latent infection in the neurons. That's where the virus will hide out and then can come back later on and produce another infection and more symptoms. Okay, and the last group 1 virus that infects people that I wanted to talk about are the pox viruses. So an example here is smallpox. Here's a picture of the smallpox virus. It has an envelope. You can see the capsid here inside. It's complex in shape because it's not helical or icosahedral. The smallpox viruses are actually very large. You can actually see them with a light microscope, so they're pretty big. And here's what it does to people. It causes these pustules all over the skin, and it ended up killing about 1/3 of the people that it would infect, so it had a pretty high mortality rate.